Hello and welcome back to the GCP-Mindset channel. Today we'll answer the question, why are control groups used in clinical trials? Control groups are essential because we are looking at statistical validity of our outcomes. A lot of times, the effect that you're seeing with your study drug may not be dependent on the drug yourself, but could be due to underlying changes within your patient population, the disease, or placebo effects. So what you need to do to increase the validity of your outcome is compare the benefits and the safety profile that you see in your trial population that is actively treated with your drug or your medical device to a comparator group. Sometimes that's placebo or a sham device, and sometimes that's an active comparator, so another treatment which is already used in this indication. And what you want to see that you're comparable, or that you're not worse, or that you're even better, that you provide additional benefits compared to this comparator arm. And that's exactly why they're enrolled in the trial, and it's really pivotal from a statistical point of view to consider them as long as feasible. They should be included in the trials. There are actually, even beyond orphan and rare diseases, there are many trials that do not have a control group. Now, whether that's good or not is a different question. At the end of the day, from an industry perspective, efficiency is always considered paramount. And frankly speaking, as patients, we should also consider this. If every trial was controlled, if every drug was developed through a series, or every device was developed through a series of controlled trials at all stages, the costs of development, the duration of development would also increase, which impacts availability of treatments for us and costs of these treatments. So we always think about it from a perspective of ethical and logistical needs also within trials. There are reasons why a control arm may not be feasible, and that can be ethical in nature. For example, oncological patients should probably only be treated with the best treatment. You cannot have a placebo in there, and hence you should ensure, if there's no active comparator, that you only have your study treatment and no control arm. That's the only ethical way you approve it. Sometimes it's not logistically feasible because it is not necessary in the core for what you're trying to assess in the trial. For example, early information on your safety. Yes, you can compare it to a control group. You don't necessarily have to, because you're only looking for safety signals maybe in healthy populations. That can also be a reason. And sometimes there are simply too few patients. This is where we get into like orphan drugs or rare diseases rather, when you have very few patients and any of these patients included in the control arm means you have less evidence for your treatment and for your safety of your treatment, and it becomes simply logistically impossible to recruit these populations in time, in a reasonable amount of effort and money. So from those perspectives, there are many examples in which a control arm is not given. If we look at the risk profile of individual products and compare drugs and devices, with devices only having a physical effect, sometimes very localized, there's also a point to be made that for the lower risk products, you may not need to go to these extremes of including logistically and monetarily and time-wise challenging control arm, especially in some of the trials, for example, after market access. There, you simply observe and you don't include a control arm. So clinical trials, in a nutshell, are very diverse, and this diversity means there are many situations in which conditionally you may not include a control arm.